Good evening, everyone, and welcome to UT Southwestern's Science Cafe. My name is Joya Lang, and I'm part of the public affairs team here at UT Southwestern. We are in our third year of hosting Science Cafe, and we're so proud to produce this series. On behalf of my colleagues, Jenny King and Charlie Thompson, as well as our guest speaker, Dr. Michael Shiloh, thank you for joining us tonight. Science Cafes are online conversations where our speakers take you on deep dives in science and healthcare topics. At UT Southwestern, we are an academic medical center, which means we bring together research, health education, and patient care into one institution. Research informs care advancements really quickly, which is a benefit to our patients and our community. This evening, we are discussing the science of coughing with Dr. Shiloh. More on him in just one moment. Before we begin, a few technical points. We are recording this program and live streaming it to our Twitter page. Please mute your microphones to help with audio clarity for everyone. We encourage you to leave your video on so we can see you, especially during the Q&A portion. We love to see our Science Cafe community. And finally, just a reminder that while we cannot answer personal medical questions, we would love to hear from you with your general questions on the topic of coughing. So any questions that you might have, feel free to type those into the chat. And with that, I am pleased to introduce our guest speaker. Dr. Michael Shiloh is an associate professor of internal medicine and microbiology at UT Southwestern Medical Center. He is an infectious disease specialist, and he runs a microbiology and immunology research lab. Dr. Shiloh sees patients at Clements University Hospital and Parkland, and he is a principal investigator of the Michael Shiloh lab. Charlie will paste his link in the chat. Please look through all of his research. It's fascinating. Dr. Shiloh, thank you so much for joining us this evening. Welcome to Science Cafe. We're so happy to have you. The virtual podium is yours.
Thank you so much, Joya. I'm going to I'm going to attempt to share my screen. Okay, fantastic. Well, again, thank you, Joya, for this, the kind introduction. And we're going to spend the next 30 minutes or so talking about cough. And my intention is to give both a historical background on cough, what it means for infectious disease and transmission of infections, and also talk a little bit about the research that's ongoing at UT Southwestern in my lab and with collaborators on how certain infections actually initiate a cough, potentially to mediate their own transmission. So today I'm really going to talk mostly about infectious disease-associated coughing. Of course, there are other causes of cough that we can certainly discuss, but today I'm going to focus on infections. So this is a very short video of an individual coughing that I obtained from my collaborator at MIT, Lydia Buriaba, who's an engineer. And you can see right away that this individual in this slow motion video is producing thousands of particles, some of which are very small and float in the air, and some of which are very large and appear to drop down very quickly. And this is going to be a topic we discuss a bit during this Science Cafe. So obviously this is an impressive slow motion video. And what I wanted to switch to next is actually a poll that we're going to launch, and we're going to ask you to vote. And the poll is, which of these infections is not spread by coughing? And you can look at the options: tuberculosis, coronavirus, measles, chickenpox, or strep throat. And we're going to give everybody about 15 or 20 seconds to vote, and then we're going to talk about the answer. Okay, so it looks like we've gotten a fair number of respondents, and the primary choice was chickenpox. So let's see what happens. Okay, so this is something that's probably not familiar to many people, except for those who've been around a long time. This is measles, and this is a young child with measles. You can see the characteristic rash. Turns out that measles is actually spread by cough. One of the symptoms of measles, other than the rash, is something called coryza, which is just a fancy word for cough. And many of us have become experts in something called the R naught, which is the transmission value of an infection. Meaning, if I have an infection and I'm the index patient, how many more people can I spread it to? Measles has the highest R naught of any known infection, more so than COVID or tuberculosis. It's roughly estimated to have an R naught of 18, which means that one individual can potentially spread the disease to 18 others, and it's spread by cough. Here's another infection. This one is also probably likely reminiscent for some people who've been around a long time. This is chickenpox. And those of you that voted for chickenpox, I'm sorry to say, didn't answer correctly. Chickenpox is actually an airborne infection. And so when people used to bring their kids over to have pox parties, and the notion was you could rub the pox on people. Yes, you can spread chickenpox that way, but actually the root of infection is primarily through the airway, and that individuals cough out the virus, and then naive individuals breathe it in. And that's why you end up with sometimes pulmonary infections with chickenpox, especially as adults, which can be very severe. So COVID, of course, can also spread through the cough. I actually thought I had another video here, but it seems like I didn't. I mean, anyway, COVID, of course, is another infection that we all know can spread by cough. About 2/3 to 3/4 of people with COVID have cough as a major symptom. This last image is not of someone coughing, but it's an unfortunate individual with advanced tuberculosis. And you're not seeing the individual cough, but what you are seeing is the characteristic signs of tuberculosis, which is significant weight loss, what we call in the medical field cachexia, but what we used to be called consumption. And I bring that up because it's been known for a long time that infections can spread in the air. And Aristotle, more than 2,000 years ago, noted why, when one comes near consumptives, that's people with tuberculosis, why does one contract their disease while one does not contract it with these other diseases? And then he said, with a phthisic, that's another word for tuberculosis. The reason is that the breath is bad and heavy. One takes the disease because there's in the air something disease producing. People knew more than 2,000 years ago, if someone was coughing and ill and they were losing weight, i.e. consumption, they were probably transmitting infection to the next person. We can look at our own country here in the United States. In the New England Journal of Medicine, about 10 years ago, they looked at a retrospective over 200 years of medicine. And this is the mortality of individuals in Boston in 1812. And you can see Boston was not a huge city then. There were 942 reported deaths. The majority of deaths were from consumption. Almost 1/4 of deaths were from consumption or tuberculosis. There were also a large number of deaths from febrile illnesses, including pulmonic fever, which is probably pneumonia and other types of airway infections. You can note here, cancer was a very small percentage, and maybe they didn't know enough to know what was cancer and what wasn't, but it was not a high percentage. And then, of course, if you look this over very closely, you can see some odd things like drinking cold water caused death, as well as teething or worms. Now, TB has been around for a very long time, and back in the 1800s, it was the leading cause of death worldwide, and it still is one of the leading causes of death. And people looked at it in art. So Edvard Munch is best known for his painting The Scream, but he also painted this picture called The Sick Child, which was his sister who was dying from tuberculosis in bed. And this is his mom, essentially grieving over her dying daughter because she was dying from tuberculosis. Cough and infection spread by cough is such a big problem that at the turn of the century, in the 19th century, there were lots and lots of cough medicines being sold around the United States. And this is in the era of quote unquote snake oil salesmen, but lots of cough medicines, Dr. Bull's cough syrup, Dr. Boshi's, they tended to include things like alcohol. This one had 4% alcohol. This one had morphine in it. This one reportedly was going to cure you in 1 minute. And it had all these things it was going to cure, like whooping cough and bronchitis and pneumonia and hemorrhage of the lung, which is bleeding lungs. There were so many of these medicines being sold with so many things like alcohol and morphine that other entrepreneurs took advantage and wrote that their cough cure positively contains no opium or any other harmful stupefying drug. So they were going back and forth. My medicine is, drug-free. These medicines have them. It was, of course, a big issue. Now, the data in the New England Journal from that article in 2012 is really deep. And I encourage people to go and sort of play with this interactive graphic. But this is a busy slide. This reports the top 10 causes of death in the United States from 1900 to 2010. And there's a lot going on here, but if you just look, if you eliminate some of the non-infectious things and you look at influenza, GI infections, and tuberculosis, you see that in 1900, they represented the number 1, 2, and 3 causes of death in the United States. And if you're paying close attention, you see this big sharp peak of influenza in 1918, which was the classic 1918 influenza pandemic that both ended World War I and also initiated the Roaring Twenties and many other global health altering events. You can see these infections and deaths from them have declined as public health and medical care have improved, which allows us to live longer and then develop the disease of chronicity and the diseases of aging, namely cancer and heart disease. But so we think, okay, so now we're free of tuberculosis, and at least in Western countries, we tend to be free of tuberculosis and other severe infections. Of course, COVID has proven otherwise. But we're not that far away from developing tuberculosis outbreaks, even in established countries. So this is from the front page of the New York Times back in 2018. Venezuela used to be a very well-established country. They had excellent healthcare, and then their economy and government collapsed. And these are all prisoners in a Venezuelan prison. And there was a massive outbreak of tuberculosis in the prison. And you can see people, this again in 2018, before COVID, trying to cover their face with masks to prevent catching tuberculosis. And so we know that TB spreads in prisons around the world. And all it takes is the social structure of a country to break down for tuberculosis to come back. In Africa, tuberculosis is so significant that when people go to the healthcare clinic, and in particular, if they have things like HIV or cancer, you don't want them coming into contact with people with TB. So my colleague, Dr. Mumford, who used to work here at UT Southwestern, sent me this photo one time of the so-called coughing spot, where people who actually have a cough go somewhere else than the people who are coming to see the doctor for, say, their HIV medicines. The New York Times in 2003 was trying to make the same point. So many of you we're all sort of suffering now from the pandemic fatigue of COVID or SARS-CoV-2, but in 2003, we had the original SARS. And this graphic was produced to make the point that while the original SARS was bad, it killed about 350 people. Tuberculosis in 1 year kills 2 million people. And so this year, if we produce this infographic with COVID, we've unfortunately lost more than 1 million people in the United States and many millions worldwide. So COVID has overtaken tuberculosis temporarily, but this hasn't gone away. So the point of this is to say that a lot of our airborne infections, tuberculosis, measles, pneumonia, et cetera, are still here. We just have kind of put them aside because they either don't exist in our communities or because we are right now focused on a global pandemic. But you can see TB, measles, influenza, they don't even talk about pneumonia here. So this prompts me to bring up the second poll question, which is coughing good for you? True or false? Coughing is good for you or helpful when you have a respiratory tract infection. Well, it looks like everybody believes that coughing is good for you. And this was a bit of a trick question because, quite honestly, we don't know the answer to this. It certainly is common sense that if you have mucus and you can't breathe very well, that coughing and expectorating that mucus will help you breathe better. And maybe it's eliminating some of the harmful organisms that are in your respiratory tract at the time of infection. On the other hand, we really don't truly know whether suppressing the cough is good or bad for you. So all of us have at one point or another either taken or administered to a family member or child a remedy that we bought at the pharmacy. And some of them have cough suppressants in them, like dextromethorphan, or with a prescription, you get codeine. Others are expectorants. They're meant to help you cough. And so it kind of goes both ways. Do we want to cough the stuff out? Is it beneficial? It certainly isn't beneficial from the standpoint of transmission. We still don't even know whether it's good or bad for you when you have an infection. So this was a bit of a trick question, and I apologize if people are annoyed. But it just made the point that we common sense think that it is. And so it also allowed me to introduce the general physiology of cough. So again, from the New England Journal from a few years ago, what happens when you have a cough, especially one that's stimulated by a noxious agent, is there are nerves that innervate your airway and lungs. And they, on the edges of the nerves, have receptors shown here, like P2X3 and TRPV1. These are receptors, these are proteins that detect noxious materials or sometimes detect molecules produced within the airway itself. The classic one that we look at is TRPV1, which is actually the receptor for capsaicin, which is hot pepper. So if you're exposed to pepper spray, the reason you feel ill, the reason you want to cough is because of the engagement of capsaicin with this TRPV1 receptor. And indeed, this past year, the Nobel Prize for Physiology and Medicine was awarded to the discoverers of TRPV1 as the receptor for capsaicin. Once that signal is detected by the nerves in the lung, it sends a signal to the brain, and then the reflex is initiated, leading to first closure of your glottis, an increase in pressure in your lungs, and then release of the glottis, which then releases the cough reflex. And you can see that in the brain stem, in the brain, there are receptors too that are initiating the cough response. And the two that I'm focusing on are the NMDA receptor, which is actually the receptor that is engaged by the medicine dextromethorphan, which is the medicine that's in Robitussin, and the opioid receptor, which is the medicine that we target with codeine and morphine. This one tends to make you mostly sleepy, and then you cough less. But keep in mind that the signal comes down back here through the so-called vagus nerve, which is a very important nerve, and we'll return to that in a minute. But the other nerves that are involved are these jugular fibers and the nodose fibers, which we'll also talk about very briefly. So how do we know that this works? So this is an example of a human study. So about 30 years ago, some human volunteers were exposed either to capsaicin, which is from hot pepper, or citric acid. They were put in a plethysmography chamber, and that's basically a large, in human beings, that is a large phone booth sized chamber, and then you measure the coughs as you expose them to increasing concentrations of hot pepper. And you can see that at increasing concentrations of the hot pepper, people are coughing a lot, 15 or 20 times. They don't have the time scale here, but this is within 10 minutes. In parallel, if they gave the same individuals something called citric acid, which is just an acid stimulus, similar to what you would experience if you regurgitated your stomach acid and then it went in your lungs, citric acid also induces coughing, but not as robustly or profoundly as with capsaicin. So we can return now to this cough reflex and just show you a little bit about what's going on. So I told you that this vagus nerve is the primary nerve that engages the cough reflex. And the vagus nerve serves a very important purpose. It actually innervates a large number of your visceral organs, your lungs, your heart, your liver, your pancreas, the stomach, and small intestines. And it reports on the activity of various signals from the visceral organs back up through towards the brain. And in doing so, it passes through something called the nodose ganglia that is in your neck. And then the signals then come back through the vagus nerve to affect organ activity. There are also a few nerves that live in your spinal cord that go through something called the dorsal root ganglia, which is a tiny little clump of nerve bodies next to your spinal cord. And these can also innervate some of your visceral organs as well as your skin and other tissue. And this is just one representative example. This is a piece of lung, of human lung, that was cleared of all other tissue except the nervous tissue, shown here in green. And you can see these tentacles of nerves, or what we call dendrites, and it looks like the arms of an octopus. And these are reaching out into and through the lung, and all those receptors I talked about are sitting here attempting to identify if you've been exposed to a noxious stimulus. So let's return to the cough. We answered that common sense question that the cough is good for you when you have an infection, but it's certainly, if you look at this image, is probably not so good for your neighbor or your spouse lying next to you in bed. So, what I want to talk about now is, how do we know that cough actually has infectious particles in it? How do we know that it's transmitting infection? What's the difference between droplets and small particles called aerosol particles? How do we know that cough is associated with infection at all, despite, a lot of this is common sense, but as scientists, we want to be data-driven. So it turns out one of the first lines of evidence to show that people with TB actually have a cough that's responsive to treatment came from our very own Parkland Hospital more than 50 years ago. And two doctors, Robert Luden and Sharon Spohn, did something really interesting. And I put this picture up because this is right from their paper. They used this very, very sophisticated, for its time, recording equipment. And they took patients that had tuberculosis and monitored them in the hospital and recorded them coughing while they were in the hospital with their tuberculosis. And I'm not going to show you all the data from their work, but one thing that I found really interesting is, so they had 20 patients at the beginning of their work that were admitted to the hospital. And you can see some of these people were in the hospital for six or even 9 weeks. And before treatment, over 8 hours, many of them were coughing over 100 times over 8 hours. That's a lot. That's more than 10 times an hour. And then they started giving them treatment for their tuberculosis. And you can see that as treatment began, their number of coughs went down, showing a relationship between the number of bacteria in the person and the number of coughs that they were experiencing. But you can note here that even by 4 weeks, a month after treatment, at least almost a quarter of people are still coughing. In tuberculosis, this is known to be the case, that people may still be producing infectious bacteria, especially if the initial burden of infection was very high. But in other infections, say from a virus that causes you to cough, you can have the virus be coughing. The virus can go away and you can be coughing for weeks or even months after, the so-called post-infectious or post-viral cough. And that's a different kind of syndrome. The point I wanted to make here, though, is with TB treatment, coughs were reduced, suggesting a relationship between the presence of bacteria and coughing. So let's talk a little bit about the particles that are actually produced when you cough. And a lot of us are probably really curious about this because there's been lots of discussion, particularly with respect to COVID, about droplets and aerosol. How do we know that these particles are produced, and what is their very nature? And so about 90 years ago, William Wells performed some experiments, and what he concluded is that there are two types of particles produced when people cough or sneeze. One, and he gave them names, and based on their characteristics. One he called the droplet, and here he says the pathogen falls to the ground in large droplets. It doesn't go very far, and if you can block it, if you intercept the drop's path, meaning if you're the person standing in the way of the drop, you might get infected. The other way to interpret this is if you intercept the drop before you get infected, if you block the drop, say with a mask, you're much less likely to get infected. The other route is the airborne route. The pathogen gets lifted up into the air, and then these droplets then float and can move long distances. And if you inhale this mist, you can get infected. And this model, shown here at the bottom, highlights how this works. And this is a hypothetical model where you have some pathogens inside a droplet, and over time, as the droplet evaporates and desiccates, it becomes much more compact and dry, like a mote of dust. So this is just a model, but we can actually do experiments to verify this. And this experiment was performed in 2018, and these authors, they took a droplet that they were able to label with a fluorescent dye on the outside. And you can see at 95%, the numbers up here represent humidity. At 95% humidity, you have this perfect looking sphere. And this looks like almost like a balloon or like a bubble you might blow or a kid might blow from their soap, like a little soap bubble. But the minute you dehydrate it, even as low as 80% humidity, you can see this begin to contract. And so by the time you reach 40, 35, 30%, it's really this very dense particle, maybe a quarter or fifth of the size of the original one. It's these particles that end up floating in the air and then potentially sitting in the air for a very long period of time. So that's what leads to this very simplified model. This was taken from a website that was hosted by NPR, And this was around the time when Ebola was a big concern, so for those of you that remember that. And what this tried to highlight is that larger droplet particles, including those that might be produced by people with Ebola, but certainly other infections, they drop to the ground very quickly because they're big and they're heavy and they're dense. Whereas smaller particles can be produced and go very, very far, 6 feet or farther. And they use measles as an example, which is what I highlighted earlier. Part of the reason measles is so transmissible is because it can just float and drift, and people can encounter measles a far distance from the initial patient, and even delayed in time. So this person can produce a measles cough, walk away, an hour later, you can walk through their aerosol and contract measles. So that's how aerosols work. So now you know how the different particles are produced. How do we know they even have infection in them in the first place? I know it's common sense and we think that's true, but how do we know that? Now, William Wells did some experiments many years ago where he captured some of these particles and then used them to infect animals and showed that they contained viral infectivity. But we can go one farther. So about 20 years ago, a group at the NIH did the following experiment. They took individuals that had tuberculosis and they attached them to a special apparatus like this tube shown in the upper panel, and the person coughs into the tube. And when they cough, a pressure transducer detects the cough, and there's something called an Anderson sampler, which then separates the particles based on size, from small to medium to large. And then you can take those small, medium, large particles and then basically put them onto bacterial growth plates and figure out, are there bacteria in these particles? Shown here on the right are 5 different subjects that coughed into this machine. And we can look at different patients. So you can look at patient B up here in the upper corner, who only produced particles stage 6, very, very small, less than 1 micron. That is a very small particle. And so this person only produced tiny particles, but what maybe you can't see is they only produced 2 to 3 CFU. CFU stands for colony forming unit, 2 or 3 bacteria in a particle. That's not very many. On the other hand, if you look at patient E, subject E, they also produce small particles, mostly 5 and 6, so mostly less than 2 microns, but they're producing 750 bacteria per particle. That's a lot. And so these individuals are much more infectious than the individuals that produce very few particles. Now, you can also see subjects that had a wide range of particle sizes, including some very large ones. But the fact that we know that patients with tuberculosis or other aerosol infections can produce really tiny particles is what leads us to recommend the use of N95 masks for the person who needs to interact with such a patient, because the N95 mask protects you against very small particles, whereas other masks tend to protect you more from slightly larger particles. So this is a table that I created that summarizes some of the major infections and where we believe them to occur in the context of droplets versus airborne particles. And I'm not going to spend too much time here, but to say that TB, or tuberculosis, which is what we're going to talk about a little bit more, is primarily transmitted by airborne particles, whereas something like pertussis is very large and is transmitted via droplets. So whooping cough by droplets, you have to be very close to someone. So this is why it spreads in classrooms where kids are sitting face to face because they're 2 feet away from each other. Other things like influenza, rhinoviruses, and now we believe the coronaviruses can be spread by both droplets and airborne particles. And I just want to make the point, and I know I'm talking a lot about infection, I just want to make the point that cough doesn't always mean infection. So in Western countries or here in the United States, the common causes of cough are really found up here. Upper respiratory infection, especially if it's acute. So if you have a cough lasting a week, it's probably an upper respiratory infection, maybe an asthma or COPD, emphysema exacerbation, maybe pneumonia. The longer it goes, the more it becomes something a little bit more chronic. So maybe it's asthma, maybe you're taking a medication like an ACE inhibitor. You could still have whooping cough or pertussis for a while, or maybe you have gastroesophageal reflux. Now, in other countries where TB is endemic, the chronic cough becomes much more likely to be something like TB. Now, frequently, people worry about things like malignancy as a source of cough, but it's less frequent than some of these other causes. So let me take this opportunity to summarize what we've just discussed. And I created this in a software program called BioRender. And I just want to take you through the 4 steps we just talked about. Step number 1 is a person produces a cough or a sneeze, and the inflammation or maybe something that's produced by the pathogen triggers the cough. And then the longer someone's contagious, perhaps the greater the burden that they have, the more likely it is that they're going to pass the infection to their naive friend over here. Now, once the cough or sneeze occurs, you get an infection, particles become airborne, or they also can become droplets. These pathogens, they pass from person to person. They can be shed with a cough, a sneeze, sometimes singing or even talking. And then desiccation or drying out of the particles lets them float in the air. Now, it's very important for some pathogens to survive for a long period of time. And so it's to their benefit to be environmentally stable, and that prolongs their survival in the air. And there's some factors that influence that, including the tolerance of the organism to be dried out, the size of the particle, and the relative humidity. It turns out that if the air is humid, like I showed you earlier, if the air is wet, the particles are less likely to become dry, and therefore they're not going to float in the air as much, which is why humidifying the air is a way to prevent transmission from one person to another. And humidity also helps because your upper airway becomes less receptive to infection. The other thing one can do, and this is what we've been doing now in schools and hospitals, is improve or increase the ventilation in the room, removing these particles from the air. Now, the last step is when these particles reach the naive person, and some organisms like measles or pertussis or COVID have receptors in the upper airway that they attach to start infection, whereas others like TB or mycoplasma have receptors in the lung itself that leads them to cause infection. Now, all of this really tells you sort of the cycle of a cough to the spread in the air to a new person catching the infection. But what haven't we not really discussed? And what we haven't really discussed is what causes the cough in the first place. Is it just that you have some nasty gunk in your lungs? It's annoying, it's irritating. Your body says, this is annoying, I want to cough it out. Or could there be another explanation? And we hypothesized there might be another explanation, and we were led to this hypothesis by a variety of sort of stimulatory data. But 1 or 2 pieces of data I'd like to point to are some studies that were published about 10 years ago. In the first study, investigators showed that another bacteria, Staph aureus, which causes boils and skin infection, produces small molecules like these peptides that irritate nerves in the skin, what they call mechanical nociceptors or thermal nociceptors, to cause you to feel pain. So if you ever have had a skin infection or a boil and it gets red and hot and painful, it's because the bacteria themselves are producing molecules that induce this response. Similarly, there's a cousin to Mycobacterium tuberculosis called Mycobacterium ulcerans. It causes a painless ulcer. It has a name called Buruli ulcer. And in a paper published in 2014, it was shown that they produce a molecule called mycolactone that hyperpolarizes and then anesthetizes the nerves in the skin, which is why the ulcer is painless. And so when I saw this data, I thought, okay, if Staph aureus can produce molecules that make you feel pain and make you feel heat, and if Mycobacterium ulcerans can anesthetize your nerves, well, is it possible that TB produces molecules that triggers the nerves in the lung to be irritated, leading to a cough reflex? And so before I show you the data for that, I will ask my final poll question. Okay, so this has to do with animal pets. And the question is, guinea pigs, which not everybody has as a pet, do they have a cough reflex similar to humans? True or false? Okay, I think we have okay. So it looks like we have a lot of people who know about guinea pig husbandry, but at the time when I started this work, while I think the guinea pig fanciers were well aware of the guinea pig having a problem with coughing, the scientific community was less sure of that, although there were scientists who studied cough who knew that guinea pigs could cough. So those of you that voted true are correct. So what do we do? How do we identify the molecule? So what we didn't know such a molecule existed, so we wanted to ask whether we had to ask questions about whether it did or didn't. And so what we did is we grew a large amount of Mycobacterium tuberculosis in our biosafety level 3 lab. Remember, this is a very contagious organism, so we have to use special facilities. And then we extracted all the organic molecules from that bacteria. And this is just a profile of the most prominent organic molecules that we could find. And we set up 2 different assays. One was a neuronal activation assay. So we took neurons, we put them in culture, and we asked whether if we stimulated them, they could be activated. Shown here going from kind of dark to green. And I'll show you a video in a minute. The other was to establish a cough experiment with living guinea pigs, because as it turns out, guinea pigs are used as a model for human cough. And what we were able to do is work through this process and over time isolate a molecule shown here in red that was not super abundant at the outset, but we ended up identifying as a cough-inducing molecule. And I will show you just a few snippets of data for that. So let's talk about the guinea pigs first. We put them in this special whole body plethysmography chamber, which is a mouthful. And I'm going to call it a cough chamber from now on. What this does is it measures pressure and volume inside the chamber using a special pressure transducer. So when the animal breathes or sneezes or coughs, you can measure that in the animal by a computer reading. And so what this looks like is the animal's kind of sitting in the chamber, and then when it coughs, you see this big spike in pressure as the animal's coughing. And again, it's kind of breathing, and then at 5 minutes, you can see 2 more coughs. And this is automated, so the computer detects these coughs and then informs us, okay, this animal over the span of this 5 minutes coughed 3 times. So then what we did is we took guinea pigs, which are also a model for tuberculosis and have been so since Robert Koch discovered the bacteria more than 100 years ago. We infected them with TB and then measured coughing in experimentally infected animals. And what we found is that at 6 weeks after infection, when the bacterial burden was the highest it could be in these animals, that they were coughing, infected animals coughed, shown here in blue, about 12 times in 24 hours, whereas uninfected animals coughed around 5 times, which is basically what their baseline was. So not a lot of coughs, but definitely more in the infected animals than in the uninfected animals. And lastly, what we did is we took that organic extract of molecules and we were able to nebulize it, meaning we were able to squirt it into the chamber like you would an asthma inhaler and let the animals just breathe the extract, much like the humans that I told you earlier were breathing in capsaicin or citric acid. And what we found is that when we exposed these guinea pigs to citric acid, like human beings, they coughed around 12 times in 20 minutes. But when we exposed them to tuberculosis, they coughed just the extract. There's no living bacteria. This is just organic molecules. They coughed around 8 to 9 times in 20 minutes. So we were onto something. There was something in the TB extract that alone could cause the animals to cough in a very short period of time. So I just need to remind people that the types of nerves that mediate cough are either the vagus nerve that it lives in the nodose ganglia in the neck, or some spinal nerves that live in the dorsal root ganglia shown here adjacent to the spinal cord. Because now I'm going to turn to this neuronal activation assay. And I wanted to show you a really fun and cool video. Science should be cool. So we took neurons from a mouse. This is a neuronal cell line, meaning these cells grow and propagate almost like a cancerous cell. And we can grow them in the lab. And then what we do is we load them with a dye that fluoresces green when the calcium level goes up in the cell. And so neurons, when they get activated, calcium level goes up before you get the action potential, the electric spike that leads to the nervous impulse. And so we were able to expose these to our MTB extract. And what did we find? When you expose them just to the control, just whatever your molecule's in, they're clear. But when you add the TB extract, which we call the agonist, you can see they fluoresce green. And you can also measure that by the change in fluorescence. And you can see these big peaks occurring after the addition of the TB extract. So I'm not going to go through a lot of data today. We have a lot of data. You can ultimately read the paper that we published, and I'll provide the reference at the end. Here was that experiment I just showed you. You can see that when we add like the solvent that we put the extract into, there's no activity in the nerves. But when we add the TB extract, we have a lot of activity. And if you look in the blue triangles, those are individual neurons that are activated by the tuberculosis organic mixture. So a lot of scientists feel like cell lines are not the best because something wrong has happened and they're basically functional like cancer cells. So we decided to do this with 3 other different types of neurons. First, we collected dorsal root ganglion neurons from mice and showed that they respond to the TB extract just like the neuronal cell line. Next, we did something more complicated, which is to isolate neurons from the nodose ganglia. So this meant doing some surgery on the neck and finding these tiny, tiny little clumps of cells and putting them into culture. And then we found, again, that these nodose neurons are activated by tuberculosis organic molecules. And lastly, we were able to obtain dorsal root ganglion neurons from 2 human deceased donors and tested whether they also responded to the TB organic extract and showed that indeed they did. So this is definitely a characteristic of different types of neurons. And to make a very long story short, this was the molecule we identified. It's called sulfolipid 1. It is a sugar. These 2 things that are these two chair molecules is a sugar called trehalose. It's sulfated, which is why it's called sulfolipid, and it has these long lipids on it. And you can see on this graph that when we have the TB extract, like I showed you before, the neurons are activated and just pure sulfolipid. There's nothing else. There's no other molecules is also enough to make the nerves be activated. So this is the molecule that we identified that triggers the nerves. And a lot of you are probably going, I haven't looked at biochemistry or organic molecules in 30, 40, 50 years. I don't want to look at it ever again. I don't really want you to focus on this except to look at the top where it says, where you see the simple sugar. It's a disaccharide called trehalose. And the trehalose is actually a food additive that lots of companies add to food, surprisingly enough. But the bacteria, they use this trehalose and they add, using an enzyme called STF0, a sulfate to it. And I'm only pointing this out here because we can do genetic tricks in the lab where we can delete this sulfotransferase gene from Mycobacterium tuberculosis. If we do that, these bacteria don't make this molecule. They also don't make sulfolipids. So it's a way to make a TB strain that behaves just like tuberculosis, but doesn't make a single molecule, namely sulfolipid. So what we did is make that TB strain, and then we asked, does that induce coughing? And so here, remember, when we took guinea pigs and we put the wild type extract, the extract from normal TB, they cough around 10 times, these guinea pigs. If you take the same exact guinea pigs, you let them rest after they've been coughing, let them rest 2 or 3 days, then you expose them to TB extract from a strain that doesn't make the sulfolipid, there's no coughing. And then we can do another genetic trick, which is add that enzyme back, and now you have coughing again. And just to show you, this is very much similar to what these same animals were coughing with citric acid. So this is the control. So again, we concluded that without sulfolipid in the mixture, there's no coughing. And we also we then got purified sulfolipid. We exposed the guinea pigs to purified sulfolipid, shown here in yellow. And again, they were coughing quite a bit. And now you're probably thinking, okay, there's one more experiment I want to see. And I'm going to show you the experiment. So we took guinea pigs, we infected them with regular tuberculosis, the one that we were using all the time in lab, or tuberculosis that was missing the sulfolipid. And at 6 weeks after infection, you can see that those infected with regular tuberculosis coughed about 10 times, which is what we had seen in the first experiment. But then when we used the strain lacking the sulfolipid, no coughing. And again, we used our genetic trick, and the strain that had the sulfolipid added back was coughing just the same as the regular wild type tuberculosis. So that's it for the data. What we found and what we published 2 years ago actually is that when TB are in the body, they produce this molecule sulfolipid. It triggers nerves to fire, leading to a cough reflex. And so you're probably thinking, okay, that's great, and tuberculosis is a bad disease, but what does this mean? So yes, TB produces sulfolipid, which activates the neurons to fire, which leads to a cough. But I also told you a lot of other organisms are spread by cough, including coronaviruses, influenza, measles, bacteria like Bordetella and Mycoplasma. It's quite likely that they produce molecules, maybe not sulfolipid itself, but similar to sulfolipid, that do the same, that initiate the same phenomenon, namely cough and transmission. And so we're very interested in understanding whether these organisms do something similar to TB. And that's what we're working on now. We have ongoing projects to identify other molecules produced by bacteria that trigger this response. We are working to identify the receptors that are on the nerves that mediate the cough reflex, shown here. And lastly, one of the more interesting questions is, okay, so let's just say we can find the cough molecule. Does it actually mediate transmission from infected animals, shown here, to uninfected animals, shown here? Because that will prove that the cough is not, that the production of these molecules is really an evolutionarily selected mechanism to enhance the transmission of these pathogens, something that we suspect but have no proof for yet. And so hopefully I will be able to find something like a cough cure, and I put this up at the end. This is my second to last slide. This is another one of these home remedies for the cough that existed 100 years ago. This isn't Photoshop. You can find this in the National Library of Medicine, and it just so happens that it has my name. And what's interesting about this particular cocktail is that it actually had chloroform in it, shown here. So you really, if you took this, you really were knocking yourself out, and probably that's why you weren't coughing. And of course, this work can't be done without a team of people. This is actually an old photo, so I had to update it with newer people in my lab. These are the members of my lab that have or will have conducted these experiments. The main one is a student who just graduated a couple of weeks ago, Cody Rule, as well as a technician, Lexi Kint. We have collaborators at UT Southwestern, as well as UT Dallas. So definitely keeping it in the UT system. And all of this was done with the financial support of the NIH, so all of our tax dollars, as well as UT Southwestern and two foundations, the Burroughs Wellcome Fund and the Welsh Foundation. And with that, I'm happy to take questions.
Dr. Shiloh, thank you for your presentation. We would now like to get into the Q&A portion of this session. And first, we'll start with a few questions submitted ahead of time, and then we'll address questions from the chat. The first question is, I would be interested in learning if the physical process of sneezing and coughing are related, can either be triggered by the nervous system unrelated to allergies or viruses.
That's a really great question. Yes, sneezing and coughing are similar processes. They are mediated by similar nerves. And it turns out actually that sneezing produces even more particles than coughing, probably by an order of magnitude. So yes, they do share similar properties. There are unfortunately fewer models of sneeze than cough because cough is an easier physiologic mechanism to physiologic reflex to measure. So yes, they're similar. It's less well studied simply because the models are much more challenging to work with.
Thank you. The next question is, does long-term coughing of 40+ years due to environmental allergies cause other medical issues?
Yeah, it certainly could. Coughing, one of the things that I didn't mention, and there's a lot to talk about, one of the things I didn't mention is cough. If you think about it, cough generates a tremendous amount of mechanical force in your lung. And your lungs are constantly stretching and contracting when you breathe. That's a normal process. Each of us can take a deep breath, and then there's recoil in the lungs as they stretch and contract, stretch and contract. Coughing makes that expansion contraction much more violent. Imagine taking a saltwater taffy and stretching it and contracting it. Eventually, if you're stretching it, it's going to break. And so continued coughing, especially robust coughing over a long period of time, can lead to issues with emphysema and the like. It's even possible that some of the diseases that we see, like tuberculosis, that has this so-called cavitary lesion, is caused because of cough. As the intrathoracic pressures go up and down, it stretches and contracts like a balloon and creates these cavities in the lung. So long-term coughing could be a sign of something serious and could cause long-term problems. It is difficult to separate the problems that you see in the lungs from a cough from the underlying disease that's causing the cough in the first place. But the answer to that question is yes, there can be issues associated with that.
Thank you. Here's a question from the chat. What are common causes of chronic cough in otherwise healthy persons?
So the four most common causes are postnasal drip, gastroesophageal reflux disease, asthma. There's a form of asthma called cough variant asthma, and actually pertussis is the fourth cause. So people don't realize this, but in adults, if you exclude postnasal drip and if you exclude asthma and gastroesophageal reflux disease, then pertussis or whooping cough can actually occur. It turns out that the newer vaccines, the acellular pertussis vaccine, the immunity wanes over time, and people can develop pertussis, and coughing can be so severe that it can lead to post-tussive emesis. You can vomit from the cough, or you can break ribs. So there are potential causes of chronic cough that can occur in otherwise healthy people. Then you get a diagnosis of whatever the issue is, whether it's esophageal reflux or asthma or something, and then hopefully you undergo treatment, which helps with the cough. If you exclude all of that and you end up with something called idiopathic cough, there are some other medicines that people can be given, but that requires being seen by specialists like in our pulmonary or otolaryngology clinics.
Thank you. Is there a way now or in the future that we'll be able to tell the difference between infectious and non-infectious coughs?
Well, that's a fantastic question. People are working on this. There was the Bill and Melinda Gates Foundation for a long time has been supportive of sort of high-risk ideas. And one of the ideas that they supported was an iPhone app that would allow an artificial intelligence-based system to detect if someone coughs into their iPhone, whether the cough is more characteristic of an infectious or non-infectious cough, and even if it's a cough that's consistent with, say, tuberculosis versus pertussis versus something else. And so they have developed algorithms that can detect the difference between those types of coughs, and these are now being tested in a variety of centers across the world. And if I might, after this talk, post on my Twitter feed a link to some reports describing this type of technology, initially supported by the Gates Foundation and now being taken on by several software development companies.
Wow, is all I can say about that one. Technology is awesome. Okay, here's another question from our chat. Can anxiety trigger coughing?
Can anxiety trigger coughing? So it's a good question. I'm going to try to spin it a little bit and say that, so I actually was going to ask this as a true false question. Cough can be both involuntary and voluntary, right? And I think those of us, once you hear that, you think about it for a minute and you realize it's true. So if you go to a movie theater and you feel like you need to cough, or if you go to a show and it's like the big end moment and you feel like you're going to cough, you sit there, try to suppress it. So voluntarily, your brain can send signals to your body, your lungs, your throat to try to suppress the cough until you basically can't. So that's voluntary control. And so anxiety may trigger coughing through the loss of the ability to control voluntary coughing. The involuntary coughing could be something like, I just drank some water, it quote unquote went down the wrong pipe and went into my lungs. My airway says, well, water's not supposed to be coming down that way. And then you have this expulsive cough as a straight reflex that you don't control. So involuntary cough is basically a protective mechanism to prevent material from coming down the lungs, from your stomach, from your mouth, whereas the voluntary cough can potentially be suppressed or exacerbated by things like anxiety.
Thank you. Here's another question from the chat, and this has to do with covering your cough. What are your thoughts on coughing into your clothing rather than your hand or your arm?
I don't know that there's a definitive answer for this. I think there was a lot made about potential fomite transmission of COVID at the outset when people didn't know. The first thing I would say is absolutely would be valuable to cover your mouth when you cough, because you can try to reduce, not completely, but by some significant amount, the particles that you're spitting out. Same thing with a sneeze. If you can cover your mouth or nose when you sneeze, you're going to reduce the particles that are released into the air. I think that what I would say is, it's hard sometimes to know you're about to sneeze or cough, and when it happens, if you could do your best to either cover your mouth, whether it's with a handkerchief, whether it's with your hand, whether it's coughing into your elbow, you can do everything you can to kind of prevent the spread of that infection. I don't think that there's definitive evidence to argue that your hand is any worse or better than your arm or elbow or your sleeve. I think some people might speculate, well, if you just coughed on your hand and then you go and shake someone's hand, you haven't really done them a service. But I would argue if you cough on your hand, you probably want to go wash your hands pretty rigorously before you shake hands with anybody.
Thank you. Here's another question from our chat. Is there any evidence showing a link between dairy consumption and coughing? People often say that dairy isn't good when you're sick or have a cough. What does evidence show?
Wow. So I'm going to admit a little bit of ignorance to this. I've not heard that dairy consumption is something that makes coughing better or worse. I would have a hard time connecting that to a mechanism. I don't know how that might occur. I don't know if one would speculate about the lactose in there, but I've never heard that connection. It could, it could be, a old wives' tale, and there's a lot of those out there that are based on folk sort of passage of concepts over time. Some of them are quite valuable, but others, I don't think we really know whether you're supposed to do the things that are recommended. I would say if you have a cold, chicken soup is always good, but I don't think there's any evidence for that either, right? So it's like a old-fashioned version of penicillin, right? Maybe the chicken soup works because you eat hot soup and it humidifies your mouth and it kind of prevents anything from going on. I don't know, but I've learned chicken soup, so I'm going to stick with that.
Thank you for that. Okay, here's another question that was pre-submitted. What are the best methods for helping a child stop coughing at night when they're sick?
Boy, that's really tricky. A lot of the medicines that have been, I'm not a pediatrician, but a lot of the medicines that adults take for cold symptoms have not, actually a lot of them existed long before we ever did randomized studies, and they were sort of grandfathered into being used. And emerging data are that a lot of the medicines that we give, over-the-counter medicines we give to children, are probably not ideal. What I would suggest is one major thing that can work that's probably that's easy to do is to humidify the environment. The humidity loosens the mucus. It actually has been shown in other studies with other viruses that it actually, in many cases, reduces the amount of virus that's produced. It can also help reduce the transmission of viruses. So if you have two kids, one's sick, the other one's not, if the room is more humid, the non-sick child is probably less likely to catch it. If the room is dry, like in the winter when you have the heat on, versus having it be more humid in the room. And as I showed in the presentation, the humidity can make the particles be less floating in the air. They'll drop down and become, hit the ground and be less infectious. So there are not a lot of ways in childhood to do things that have been tested in any kind of rigorous study, but humidity is a very simple remedy that can help alleviate symptoms, but also help prevent transmission from individuals within households.
Great. Thank you, Dr. Shiloh. Before I close this out, do you have any final thoughts or anything else that you'd like to let the people know?
I would just say, first of all, I appreciate everybody for coming tonight. Thank you so much. The research that my lab conducts is actually supported by all of you through NIH funding, which we truly value immensely. We wouldn't be able to do some of this groundbreaking research without the support of the United States government. And while today's presentation may not be obvious of a cure for an infection, for example, or much like the vaccines for COVID, the work that we do now is foundational to work that we hopefully will do in 5 or 10 years, or that other people will build on. And so all I would say is we really appreciate the support of the community for this kind of research, and that we hope that it will someday lead to treatments or cures. But even if it doesn't, it does inform us about the basic mechanisms of disease, and we think that's very important. And so again, thank you so much for having me tonight. And I have a Twitter account. Like I said, I will post some of the comments that we just discussed on Twitter. I left my email address, my welcome communication as well. And with that, I'd like to thank everybody for coming.
Thank you so much, Dr. Shiloh, for this enlightening conversation on coughing and the causes of cough, and the fascinating research on the transmission of infection through coughing. Huge thank you to Charlie for helping run tonight's event. Really quick before we close out, I wanted to let everyone know that in 2 weeks, our next Science Cafe will be on April 28th on cancer disparities in minorities. It'll be really fascinating, so we hope you'll join us. Charlie will paste that into the chat, and if you don't catch it now, we will send an email to everyone next week. So on behalf of our faculty speakers and the Science Cafe team, as we close out this evening, we wish you good health, wellness, and a good night. Thank you for joining us. We are adjourned.
Thank you. Have a good night, everybody.